Apparently, I'm live, which is good. We confirmation of that. Um, Good afternoon and welcome to the College of Medicine's Dean's Distinguished Lecture. And today, of course, we're really delighted to welcome Dr. Raymond Deshaies, who is here with us today to present his work, Any Target, Every Time, How Multi-Specific Medicines Are Transforming Pharmacotherapy. And thank you so much, Dr. Deshaies, for being with us here today and sharing your work as well as your insights and into where drug development is going. We launched this Dean's Distinguished Lecture series in fall of 2022, post-pandemic, to highlight research topics that integrate efforts by multidisciplinary teams. As we know, in fact, much of science is now done as teams, team sport, and requires different subject matter experts, whether it be in cell biology, molecular biology, systems biology, immunology, data science, all sorts of omics, high-end imaging, etc. Given this reality, it's really essential to expose our junior faculty, our students, residents, fellows, to lectures and seminars on research topics that are of general interest that integrate multidisciplinary work and teams to solve major scientific problems and have impact. The critical objective of this lecture series has been to engage our College of Medicine and research community, exposing us to cutting edge science that involves the depth and breadth of scientific knowledge, but also to bring us together around building our successful teams, scientific teams, and new ideas how to diversify and expand our research programs at all levels. So we're just very pleased that Dr. Deshaies could be here with us today, given the work that he has done. I now get to introduce Dean Prabhakar to do a more formal introduction of our guest. Prab.
Thank you. Thank you, Dean Rosenblatt. Let me first start off by telling how really excited I am to have spent quite a bit of time with Dr. Deshaies since last night and again this morning, and his interaction with the students. Here is a man with extraordinary breadth of knowledge in areas of modern science. While he has developed this extraordinary breadth of knowledge, he has dug very deep into certain aspects of biology that has helped transform our understanding of some fundamental principles, which have led to development of a number of therapeutics and a number of startup companies. So until very recently, Dr. Deshaies served as the Senior Vice President for Discovery Research at Amgen. Prior to joining Amgen, he was a member of the professional faculty in the Division of Biology and Biological Engineering at Caltech since 1994. And he became an investigator of the Howard Hughes Medical Institute since 2000. Deshaies' lab investigates the cellular machinery that mediates protein degradation by the ubiquitin proteasome system and how this machinery regulates cellular function. While at Caltech, he founded the Proteome Exploration Laboratory at the Beckman Institute. He received his bachelor's degree in biochemistry from Cornell University and his PhD in biochemistry from UC Berkeley. Subsequently, he was a Lucille P. Markey Charitable Trust postdoctoral scholar at UCSF. His honors include a Searle Scholar Award, a Beckman Young Investigator Award, and a Burroughs Wellcome Fund New Investigator Award in the basic pharmacological sciences. Then he went on to receive a New Investigator Award in the basic pharmacological sciences, the American Society for Cell Biology, Promega Early Career Life Scientist of the Year Award in '99, and appointment as a Fellow of the American Association for the Advancement. He has not even established himself. Please notice this, and he's already getting places. And then in collaboration with Craig Cruz at Yale, he conceived the idea of using heterobifunctional small molecules referred to as PROTACs to tether cellular proteins to ubiquitin ligase, resulting in degradation of the tethered protein. This concept underlaid the launch of numerous biotechnology companies, including Arvinas, C4 Therapeutics, and Chimera. The Deshaies group also identified small molecules that inhibit targeting of substrates to the proteasome and removal of ubiquitin chains from the substrates by RPN11. In addition, in collaboration with Dr. Hugh Rosen for Scripps and Frank Shonen of University of Kansas, he discovered P97 inhibitors, DBEQ, and ML240. ML240 served as the basis for the development of CB-5083, which entered human clinical trials in 2014. In addition to his academic work, Dr. Deshaies co-founded Proteolix in 2003, which developed Kyprolis through phase 2 clinical trials. Kyprolis was approved by the FDA in 2012, and in 2011, he co-founded Cleve Biosciences. Dr. Deshaies was elected to the American Academy for Arts and Sciences in 2011, and in May 2016 to the National Academy of Sciences. In 2022, he received a Fierce Pharma named him as one of the most influential people in biopharma. In 2023, he received the Jacob and Louis Gabby Award in Biotechnology and Medicine from Brandeis University, and in 2025 was named a laureate by the Passano Foundation. And literally 2 weeks ago, he was honored by Goethe University in Germany for his lifelong contribution. We couldn't really have a more distinguished scientist amongst Ray. Welcome to Chicago, and podium is yours.
All right, Prab, thank you so much for the effusive introduction. It's a pleasure to be here. I'm feeling a little bit addled right now. It's always wonderful to be able to visit with colleagues, especially to visit with students. And I just spent some time with students because, they're really the future of science. And so with that, is this thing operational, do you know? Away from yourself. Okay. All right. Now I could walk around. So I'm going to give a talk that's probably quite different from the talks you're accustomed to. And, it's going to focus less on data and more on, what I feel I've learned in working in the biopharmaceutical industry from 2017 through 2025, and where I think the field is going. And I like to start with this slide. This puts in context of how I think about the history of the biotechnology or biopharmaceutical industry. And so, the lesson or the thesis of my talk today is that we are in the middle of what I'm calling the fourth transformative wave in the history of that industry. And that industry, at least in the way I tally it up, really has its beginnings in 1899 when Bayer, which arose out of the German dye industry, when they essentially identified aspirin as being a component of willow bark. People have been chewing on willow bark when they had toothaches going back thousands of years. They figured out the active component in willow bark was salicylic acid, essentially aspirin, and they then synthesized that molecule, they put it into a pill, and then they marketed the pill, aspirin, Bayer aspirin. And that's 1899, and that's really, for me, the founding of this industry. Now, so that's wave number one. There were then subsequent waves. The first one took 70 years to happen. The first 70 years, it was the playbook that Bayer had developed, which was take natural substances, figure out their chemical identity, and then, develop them through a combination of chemistry and pharmacology, and make it into a pill that you could then use to treat, some ailment. And that continued for 70 years until in the 1970s and 80s, the combination of knowledge emerging from biochemistry and molecular biology enabled a different approach where you could formulate a therapeutic hypothesis up front. So let's say, I want to reduce cholesterol because there's evidence that cholesterol contributes to heart attacks. And so we knew from biochemistry that the rate-limiting enzyme in cholesterol biosynthesis is H-MG-CoA reductase. So it stood to reason that if you inhibited that enzyme, you would reduce cholesterol, and that would reduce the incidence of heart attacks. And that turned out to be true. That led to the statins. Countless other examples of the emergence of knowledge from biochemistry leading to drugs that many of which are still taken today by millions of people, I'm sure including many people in this room. Now, that remained the same into the 80s when you had the emergence of the third wave, which was really the emergence of biologic drugs coming out of advances in molecular biology. And that gave rise to, for example, the antibody drugs now that have become so common. But still, that was operating on this principle of rational drug design, where you identify, you formulate a therapeutic hypothesis, you identify a target, you make a molecule that addresses that target in the desired way. So now I'm arguing we're on the precipice. I see. Oh, okay. We're on the precipice of this fourth wave, which is the emergence of what I'm calling multi-specific medicines. And I'll have more to say about that on the next slide. So again, and I've just been through this, if you think about all the medicines that have come so far in the first, say, 100 years or so of the industry, they work all in pretty much the same general conceptual way, where you have a target, you have a molecule. The molecule engages the target physically, and when it engages the target, it either increases the activity of the target or it decreases the activity of the target. And that's true whether it's a small molecule like aspirin binding to cyclooxygenase. It's true if it's an agonist protein like erythropoietin binding to the erythropoietin receptor, or an antagonist protein like the antibody panitumumab, Vectibix, binding to and inhibiting the EGF receptor. Now, multispecifics are radically different in that now you have molecules that instead of binding to one thing and altering its activity, are binding to two things. And I'll walk you through in a few slides why I think that's transformative. But first, I'm going to cover a couple of slides about why I think these things are emerging. Why are multispecifics? Why is this wave emerging now? And the first point is that multispecifics, by binding more than one thing, enable us as drug developers to surmount biological barriers that impede the action of medicine. Now, I'll go through those barriers. The first is persistence. So for a drug to be effective, it needs to persist in the body for a while. And the problem is your body has mechanisms to rapidly clear substances taken in from the outside. And one thing you can do with a multispecific is you can make a molecule that engages other mechanisms that are there to ensure the persistence of molecules in the body. And so a key mechanism is FcRn, which enables recycling of antibodies and albumin that enter cells through pinocytosis. And so by taking the Fc domain and attaching a drug to that domain, be it a small molecule or a protein drug, you can engage this FcRn system and dramatically expand the half-life of a molecule. And for example, this was absolutely critical for the GLP-1s because GLP-1 has a half-life of 2 minutes. And the GLP-1 drugs on the market are conjugated with a lipid that binds albumin that allows you to do a once-a-week injection. Otherwise, you'd have to be injecting GLP-1 like literally every 10 minutes to have any clinical benefit. The second thing you can get with multispecificity is you could address multiple biologic axes with one molecule. And, for anyone who's done knockouts in their career, and I did a few knockouts myself when I was working at the bench, you realize that most important biological pathways and mechanisms have some redundancy built into them. And so you can knock one gene out and have no effect, and now you knock out a second gene and you have a dramatic effect. And the same is true with drugs, and making multi-specific allows you to address multiple axes at the same time. And I've just given a couple of examples. Tirzepatide, of course, is Lilly's Zepbound and Mounjaro, and it's addressing two different axes that regulate mass accumulation in humans, GLP-1 and GIP. The third thing you could achieve with multispecificity is to control the therapeutic index of a drug. Now, you can make the perfect drug, right? It binds your target and only your target and no other target. And that drug is still almost certain to have side effect. And the reason comes from just thinking about human biology, right? We are these incredibly complex machines, but there's literally only about 20,000 genes that comprise us. And the way we get around that genomic economy, if you will, is that the same gene products are used again and again and again in different tissues at different stages of development, often to do the same thing or something quite similar, sometimes to do something very different, like lens crystalline, for example, which can function as a chaperone as well as the key protein of the lens in your eye. Now, multispecificity allows us to have a drug where one component of the drug is literally like a zip code that brings the drug to where we want it to act, and the other component of the drug carries out the actual pharmacological action. And you see this, all the siRNA drugs now that are being approved have this principle. They all, so far, all the most recent ones work in the liver, and they get to the liver by a sugar molecule that binds a receptor on the liver. Same is true for antibody drug conjugates, one of which in particular is transforming the care of women with HER2-positive breast cancer. Finally, and this is the category I'll spend the most time on, this is the category I think is most exciting in multispecific drugs, where you have a drug now that's no longer even the pharmacological agent, so to speak. The drug is a matchmaker, it's a tether, it's a linker, it's an adapter that binds a target that you want to modify the activity of, and at the same time, it binds a natural mechanism that pre-exists in the body, and it's that natural mechanism that actually carries out the pharmacological action, not the drug molecule itself. So to me, this is the most radical break with the past 120 years of thinking in the biopharmaceutical industry, where the drug is no longer the principal agent of action. The principal agent of action already resides in the body. And we see multiple examples of these emerging. T-cell engagers, I'll have a little bit more to say about them. Degrader molecules like Protax and molecular glues. Emicizumab is a molecule used to treat hemophilia. Okay, the other thing I think that's really driving this emergence of multi-specific drugs, besides these biological imperatives I've just described, is that there's a paucity of real high-quality novel targets. And that's driving, an intense, some would say insane, competition on a small set of validated targets. And so we need new ways, we need new targets, number one. But what multispecificity offers us is it offers us a way to go against that same set of targets that we know about and to capture new value out of those targets. So an important lesson I'd like you to leave with, one thing to remember, is that a multispecific drug, a bispecific, let's say, it's not twice as good as a monospecific. It's literally in a completely different league, right? A multispecific can often, usually, do things that you simply can't achieve with the monospecific. If you were using GLP-1 as a drug, you could never use it as an obesity drug because you could never have a drug that you would have to inject every 10 minutes, right? Just as a trivial example. Multi-specificity is not a quantitative differential, it's a qualitative differential, okay? This just shows the competition point I just made. And whoops. Okay. Wrong button. All right. So what this shows is a graph of targets where number one is currently the most intense competitive target in the industry. Number 25 is the 25th most competed on target. And the number of programs for those targets is graphed. And this shows in 2000, in 2010, and in 2020. And there's two take-homes from this graph. The first is that competitive intensity is increasing. And I would say in 2025, it's increased even dramatically since 2020. So this data is out of date. The second thing is, if you look in 2020, for example, even the 25th most popular program in the industry looks like it has about 8 competitors working on it. And the most popular target has nearly 60. And just thinking about this from a pragmatic commercial perspective, it's great to be first to market. You get first mover advantage. If it's a big therapeutic area, being second to market or even third to market is okay. But fourth to market, you're probably going to be out of luck, except in some very rare instances, or you have a molecule that is just way better than the first 3. And so usually companies will bow out if they realize they're not going to be first or second or maybe third. And so, when you look at the 25th most popular target having 8 programs, you could infer that there's just an enormous amount of effort that is duplicated and going to end up being wasteful. And so, again, this is an argument for how multi-specificity, in the absence of new targets, there will be new targets, but in the absence of new targets, we can still capture a lot of value from targets that we know are validated. Now, I'm going to talk mostly from here on about that last category, about molecules that work by inducing proximity, these matchmaker molecules. And as I've already described, these molecules bring together a target with an effector. You could think of the drug as having 2 hands, one hand that binds the target, another hand that binds the effector, and in so doing, it induces proximity between the 2 molecules. Now, this is most commonly being used now to induce degradation of target proteins by linking them to ubiquitin ligases, but the idea is very general, right? That just happens to be the area that's received the most attention currently among startup companies. But the idea is extremely general, and it can be used to activate, to inactivate, to change the localization, any effector function you could think of in biology that you could bring to bear on a target. It's also very general in terms of therapeutic area where you could apply it, and also in terms of modality. You can do this with small molecules, you can do it with biologics, you can do it with hybrid modalities. And finally, it's general in terms of the nature of the target itself. The target could be a protein, as is the case with protaxin degraders, but it could also be a cell. And the same for the effector. It can be an enzyme, it can be a cell. In fact, the area where the most progress has been made with these induced proximity molecules is precisely with cells that target other cells, T-cell engagers. And this just shows a list of prospectively developed induced proximity molecules that have been approved to date. There's only 15 of these that have been prospectively developed and approved to date that are listed here. So this is still very, very early days, especially when you consider that every year, somewhere between 30 and 55 new drugs get approved by the FDA. So This is really just very, very early days. Now, a couple of things I'd like you to note of those 15. First of all, most of them, I think 11 out of the 15 are what are called T-cell engagers. So they're molecules that engage an antigen on a cancer cell and the T-cell receptor, bringing and activating a T-cell adjacent to a cancer cell so the T-cell kills the cancer cell. And as I said, there's 11 out of the 15 are in that class. And that's the class where there's really been the most success to date. But it's not all T-cell engagers. So there's also risdiplam, which is used against a neurological disease. And there's a molecule used in hematology there, emicizumab for hemophilia. So these molecules are being approved in different indications. The other thing I'd like you to know is this yellow box here. This emphasizes that of these 15, most of them, I think 11 out of the 15 again, have been approved since 2022, right? So this is a wave that's just beginning to build. Now, as I think already emphasized, these approvals span modalities, so small molecules and biologics, and they span different therapeutic areas. So very broad, very general. Now, for the rest of the talk, I'm going to spend a good chunk of the rest of the talk. I'm going to talk about degraders in particular, and I have to give you a little bit of background on those. So degraders work by linking a target with an enzyme of the ubiquitin proteasome system. And the ubiquitin proteasome system is a system that's found pretty much in all cells in our body, and it's a great effector for the reasons that I'll lay out on this slide. So the way the system works is you have a protein ubiquitin that gets physically conjugated to target proteins. That's a multi-step process that involves enzymes known as E1, E2, and E3. The key ones for this conversation are the E2 and E3, which come together and form a ubiquitin ligase complex that actually transfers ubiquitin onto the substrate. And once the substrate is marked with ubiquitin, there are different possible outcomes, but one of the major outcomes is that the substrate is recognized by the proteasome, a compartmentalized protease in the cytosol and nucleus, and is degraded. Now, an important feature of this system is its generality. So one reason proteins are degraded is if there's some defect in the protein. And literally every protein encoded in the genome is subject to this mechanism because every protein, as it's being expressed, to some degree will suffer a mistranslation event or a misfolding event. And those proteins then need to be eliminated from the cell by quality control mechanisms. And those quality control mechanisms involve the ubiquitin system. So that's a key point. This system is capable of eliminating every protein encoded in the genome through this quality control function. The second important point is that this system has exquisite specificity. So in addition to being used for quality control, it's used for regulation. So there's many biological processes where the process is under negative regulation by some inhibitor protein. And the way you trigger the process is you degrade the inhibitor. And the first example of this was cyclin, which mitotic cyclin blocks the exit from mitosis by keeping the mitotic cyclin kinase on. And to exit mitosis, ubiquitin is attached to the cyclin, and the cyclin is degraded by the proteasome. Now, that specificity comes from the fact that you have over 500 of these E3 enzymes encoded in the human genome. And these are the enzymes that confer substrate specificity, so they recognize the substrate, and they also confer regulation, so when the substrate is degraded in time and in space. And so these two combinations, the ability to attack virtually any protein that's encoded in the human genome and the exquisite specificity make for an incredibly powerful effector system. Now, what got me to thinking about using this as a way to affect pharmacology was a result out of Peter Howley's lab, like 35 years ago now, in 1993. He was studying human papillomavirus, and he was studying in particular his protein E6. And there are human papillomaviruses that cause cervical cancer, and they carry an oncogenic form of this E6 protein. And what he found that protein does is it binds the p53 protein that all of you, I'm sure, have heard about more times than you care to recall, the guardian of the genome. Well, p53 represses the replication of HPV. So for HPV to replicate, it needs to somehow circumvent the inhibitory effect of p53. And the way it does this is HPV E6 binds p53, and at the same time, it binds a cellular protein that Howley called E6AP, E6-associated protein, which is one of these E3 enzymes. And in doing so, it results in the rapid ubiquitination of p53 and its destruction. Now, p53 is normally a substrate for the ubiquitin pathway, and it's a short-lived protein. But in the presence of E6, it is so short-lived that you can no longer detect it by Western blot. I don't care how sensitive your antibody is. It is simply no longer detectable. So that result really attracted my attention because viruses in general are very stingy with their allocation of nucleotides, right? And they do everything possible to limit their coding size. And so if the virus is devoting coding capacity to a protein to perform this function, suggested to me that this is potentially a very effective way of the virus to essentially commandeer the physiology of a cell. And if it's good enough for the virus, maybe it should be good enough for us as humans as well. And it turns out it's not just HPV that does this. This is a list of other viruses that have been shown to encode similar proteins that act as molecular bridges between ubiquitin ligases and a cellular factor that restricts the virus life cycle. And this is not a complete list. There are many, many examples. This is a common theme in viral biology of commandeering the ubiquitin proteasome system. And so it was this realization, one of the impetuses for me to think, gee, this could be a really, really effective way to think about commandeering cells for the purpose of curing disease. Now, right around that time where I first started thinking about this, I met Craig Crews and a professor at Yale. We both had the same fellowship, a Burroughs Wellcome Young Investigator Award. We were at a meeting that they held for the fellows. And as Craig points out, his last name is C and mine is D, and we had a poster session. So our posters were right near each other. And we got to talking, and Craig was coming up with ways of using the yeast 2 hybrid system, instead turning it into a 3 hybrid system to look for small molecules that would link the DNA binding domain and activation domain reporters that you use in the 2 hybrid system. And where the small molecules would link together whatever protein domains that were fused in the 2 hybrid system. So looking for things that would induce proximity. So Craig is thinking about inducing proximity. I was thinking about substrate specificity in the ubiquitin system. And it was, it was only a matter of time that we stumbled on the thought of, gee, can we use the technology you have in your lab, Craig, to redirect substrates at will to a ubiquitin ligase to affect their ubiquitination and degradation? And this was in 1998. A year earlier, my lab had discovered the first of the Cullen ring ligases, SCF, CDC4 in yeast. So my lab was working on these SCF enzymes. We had all the biochemistry set up in my lab. A ligand, a peptide ligand for one of the SCF enzymes had literally just been reported that year. And so, the idea was, let's take that peptide ligand for the SCF enzyme, SCF beta TRCP, and let's hook it up to a ligand against a protein that Craig's lab had discovered was the target of that particular ligand. The ligand is known as ovolysin, and the target is methionine aminopeptidase. And then that should link methionine aminopeptidase to the ubiquitin ligase. We should get ubiquitination and degradation of the substrate. And so, that was our Albert's textbook drawing. And so we then set out to test this. And here's the ovolysin molecule that Craig's lab had deorphanized, attached the linker to it, and then to the linker attached peptide ligands for this beta TRCP ubiquitin ligase. And as I said, my lab had all the biochemistry for monitoring that ligase. And this shows that when we mixed everything together, we saw a smear, a high molecular weight smear. If we used the chain terminating form of ubiquitin, we collapsed that smear into a single band above the MedAP2. And if we leave out any of the components from the system, then we don't see the smear. So this smear was ubiquitination of the MedAP2 that was being driven by the molecule we made. We called it PROTAC1 for proteolysis targeting chimeric molecule. I think we were a little optimistic. We were saying, well, maybe there's going to be a second and a third molecule, so let's call this PROTAC1. And not only did it work in triggering ubiquitination, but this is an experiment done in frog egg extract. And these are extracts that can reconstitute the entire cell cycle. When we add our MedAP2 to that extract in the presence of the drug, there's a shift in molecular weight because the PROTAC we started with is a covalent molecule, covalent with respect to MedAP2. When we incubate it in the frog egg extract, you can see the upper band goes away. When we inhibit the proteasome with either of these two inhibitors, we can stem most of that loss. And so this indicated not only can we ectopically target MedAP2 to this SCF ubiquitin ligase, we can induce its ubiquitination and its degradation. And we went on to show that for multiple other substrates, establishing proof of concept that one could indeed retarget things to these ubiquitin ligases. Now, over the next few years, there were two subsequent developments that I think were really important, important sort of psychologically and conceptually. And the first was this demonstration by Ning Zheng's lab at University of Washington that this idea of using a small molecule to link a ubiquitin ligase to a target is actually something that, besides the viral example where the viruses use proteins, that in nature, plants have exploited this as a key signaling mechanism. That is how auxin, which is probably the most famous of the hormones in plants, that's how auxin works. It links a ubiquitin ligase, one of these SCF ligases, with repressor, transcriptional repressor proteins by filling a cavity that would normally exist at the interface. So the repressor and ligase normally cannot interact stably. If the auxin is there, it fills that cavity, you have stable interaction, and now the repressor is degraded. So plants not only use this for auxin signaling, but also gibberellin and jasmonate signaling as well. So plants have repeatedly turned to this as a key mechanism for hormone signaling. The next discovery was in 2014 when a combination of labs, Ben Ebert, Bill Kalin, and Chopra at Celgene, and the other two at Dana-Farber, they were studying what at the time was the second best-selling drug in the world by revenue, $10 billion a year. This drug is lenalidomide, known as Revlimid, the commercial name. And lenalidomide is used, and it's still used. It's frontline therapy for multiple myeloma, blood cell cancer. And what was amazing is that Celgene was selling $10 billion a year of this stuff, and it was not known how it worked. The mechanism of action was not known, but it worked. It was the frontline therapy in multiple myeloma. Well, what Chopra, Kalin, and Ebert showed is that lenalidomide essentially bridges two transcription factors, IKZF1 and IKZF3, to a ubiquitin ligase known as CRL4 CRBN, another one of these Cullen ring ligases. And in so doing, the IKZF proteins are ubiquitinated and degraded. So this was incredible for two reasons. Number one, literally the second best-selling drug in the world at that time is acting by this PROTAC-type mechanism. Number two, if you went to drug developers in 2014 and said, I want you to make a drug against a transcription factor, they would have laughed you out of the room, right? They are undruggable targets. Yet the second best drug in the world, second best-selling drug, was targeting a transcription factor. So this launched an enormous amount. The confluence of these results launched an enormous amount of interest. Since then, and these are data that Beacon Intelligence helped me collect, 100 startups, 80 clinical programs ongoing, 10 billion plus in venture capital raised. Almost pretty much every major biopharmaceutical company has a program working in this space. This is just startups in terms of the numbers of venture capital. So billions and billions of dollars have been invested in this approach. Now, where do things stand? As I said, there's 80 clinical programs. They all work. Remember I said there's 500 of these E3 enzymes? These 80 programs are all using just 5 of those. Right? This is still very early days. There's a lot remains unexplored here. 35 different targets. If you recall from the ubiquitin slide, I said an important aspect of this pathway is its generality, and you could see that here. This is not restricted to particular types of targets. I mean, they have to be in the cytoplasm or nucleus, because that's where ubiquitin system and proteasome are, but they can come from many different families of proteins. Now, furthest along, we have a couple of marketed products. These are selective estrogen receptor degraders. And the mechanism of this is just now being worked out, but they link, again, the estrogen receptor to enzymes of the ubiquitin system to result in degradation. There's multiple of those in phase 3. There's a PROTAC from Craig Cruz's company, vepdegestrant, that's just completed phase 3. And there's several next generation versions of lenalidomide that are also making their way through phase 3 development. So lots of action in this space, and still early days, but I think we can expect to see many medicines emerge in the future. Now, I think I'm going to skip this slide, and I'm going to skip that slide. And I'm going to finish off by talking about an area. I just wrote about this in August. I published a review in Science. And it's really a call to arms for a new way of thinking about drug development. So I've been talking so far about induced proximity, which is bringing two things that don't normally come together. What I'm going to talk about now in the last part of my talk is what I'm calling sustained proximity. So taking an interaction that's physiological, that naturally occurs in a cell, and then reducing the off rate, using a molecule that I'm going to call LOCTAC for locking things together. Now, you could think about proximity drugs in two broad categories, as I just laid out. You can induce proximity of things that don't normally physiologically interact, and that would be PROTACs and much of the other effort going on in this space right now. Or, as I just said, you can take things that interact and reduce their off rate to sustain proximity and have a complex stay around for a much longer period of time. Now, that involves reducing off rate. Now, up to now, I'll submit that almost all rational drug discovery, which dominates 98% of drug discovery, almost all rational drug discovery involves mechanisms that reduce on rate. And so I've drawn 4 examples on this slide here. So you can either have, in red are the inhibitors, an inhibitor of an enzyme reaction that binds in the active site, an inhibitor of a protein-protein interaction that binds where the proteins dock, an inhibitor of a receptor-ligand interaction that either binds a receptor or in the case of an antibody binds the ligand. These all affect on rate. They're on rate inhibitors. I'm talking about something fundamentally different, attacking off rate instead of on rate. And you can imagine different ways of doing that. You can imagine something that's like the auxin example. You fill a cavity in a protein-protein interface to give you more surface area to reduce the off rate. You could think about extending the surface area of a molecular interaction by binding adjacent to the interface, again, adding a little bit of additional energy. Or you could think of something that acts more like a staple that binds distal to the interface, but simultaneously contacts the 2 binding partners to essentially staple them together. Now, this concept is very general. It could be a small molecule. It could be a complex synthetic, like a circular peptide. It can be a biologic. It could be a hybrid modality, not restricted in terms of modality. Now, when I was writing the review, I went through the literature and found, I think it's about 50 examples of molecules that fit this definition of what I'm calling a LOCTAC. Now, some of these are approved molecules. You have the molecules that stabilize the transthyretin homotetramer, for example, that are approved. But many of these molecules are not approved. They're molecules that are in various stages of development. But almost all of these were not developed with the LOCTAC concept. So that they work by this mechanism was figured out after the fact, right? They weren't prospectively developed. And so, you know, what I'm advocating for here is to actively think about prospective development of molecules that make use of this mechanism. Now, I'm just going to work through a couple of examples. I think it's kind of intuitive that if you have an interaction and it's unstable and you stabilize it, you're going to get more, right? It's going to be agonistic. And in fact, in some cases, that's true. Here, of the examples that I describe in the review, I'll just talk about this one here. This is a program that they had at Lilly. It's based on the idea that GLP-1, one of the first things that happens is it gets clipped by dipeptidyl aminopeptidase. You remove the 2 N-terminal amino acids. Those amino acids are part of the binding site in the GLP-1 receptor. So when they're clipped, the binding site is empty. And their idea was to fill it with this molecule, this red molecule here, which would interact with the receptor and the clipped GLP-1, stabilizing both to form a ternary complex. And in fact, they found such a molecule. And normally the clipped GLP-1 is essentially inactive. This increases its signaling by over 1,000-fold. Okay, so this is an example of a LOCTAC that essentially acts in an agonistic way. And I think it's really interesting because if normally you take an agonistic drug, that drug is going to diffuse throughout your body and it's going to activate the receptor everywhere all the time. And you don't have to be a sophisticated biologist to realize that that can potentially be a problem because there's some tissues where you probably don't want to activate that receptor. And in many cases, you certainly might not want to activate it all the time. In this case, you're piggybacking on the natural ligand, right? And so you should activate the receptor primarily in the time and place where the natural ligand is and just get more bang for the buck every time the natural ligand binds, right? And have extended duration of signaling. So potentially much better tolerated. Now, even though it's natural to think if you stabilize 2 things, you're going to get more, depending on the biology, a LOCTAC can also be an antagonist. And a couple of examples, I'm going to skip that one in the interest of time, This one is just intuitively a little more obvious. So Amgen had a program to get an inhibitor of kinesin, KIF18A. And kinesins walk along microtubules. And the walking is governed by cycles of ATP hydrolysis and release, and then release of the ADP and rebinding of ATP. And so if you think about it, to walk on a microtubule, you have an on rate and off rate, an on rate and off rate, an on rate and off rate. You have to have coordinated consecutive cycles of K on, K off. And so you could potentially inhibit kinesin by blocking either K on or K off. And it turns out a drug that we had in development at the time, civilnicib, that we've now out-licensed to a company, Velastra, that's continuing to develop it. When we did the crystal structure of that, we found that it sits at the interface between the head domain of kinesin and the microtubule, contacting the head of kinesin and the microtubule, and essentially acting like a huge wad of gum on a New York sidewalk, right? Where kinesin steps down and it can't move because it's squashed down this wad of gum or this big dab of glue, and it's stuck on the microtubule. Now, the one area where I think this could potentially be most interesting is transcription. Again, getting to the point I made earlier, transcription factor is considered to be among the most undruggable of the undruggable proteins. But if you think about it, a transcription factor has a huge job. And that's illustrated just by some quantitative considerations. So MYC recognizes a 6-mer, CAC-GTG. You can calculate that in the 6 billion bases of the human genome, there are 1.5 million CAC-GTG sites. Now, there's only 33,000 copies of MYC in an ICR90 fibroblast. So there's 50 times as many potential sites as there are copies of MYC. And even worse, how many MYC target genes do you think there are? Maybe 1,000, 1,500? Okay, so you literally have a 1,000-fold excess of potential sites in the genome compared to relevant promoters. So a huge task for MYC or any transcription factor, it is asked to sort through an enormous amount of irrelevant DNA in order to find the relevant DNA. It does not have a GPS, okay? Now, people doing single molecule tracking have put some numbers on this. This is for 4 different transcription factors out of a paper from Gordon Hager's lab. And what you see is the median time for a binding event in a cell is 3 seconds or less. 90% of binding events have dwell times of 35 seconds or less. And this is the way it has to be. In order to find a target, transcription factor has to dissociate rapidly. And so instead of trying to block the association, maybe a more productive way would simply be to block the dissociation and make binding even higher affinity than it already is. And in fact, there's a publication that came out about a year ago on FOXA1 from Michael Erb's lab that shows that FOXA1 normally binds this consensus, but upstream of it, there's a weaker consensus here that has a lot of variability in it. And that weaker consensus can determine the binding affinity of FOXA1, where if it conforms to it, it has a higher affinity, like here, and if it doesn't, it has a lower affinity. And he found a molecule that attaches to FOXA1 and contacts the DNA such that it eliminates any preference for that upstream triplet nucleotide sequence. And so that leads to a partial redistribution of FOXA1. It starts binding sites that it normally did not bind before. Those are in red. And it is dissociated from sites in green that it normally used to bind. And so this is a primary screening hit. These are still early days. But it shows that by increasing the affinity, you can decrease the specificity and redirect the occupancy of a transcription factor. And so with that, I'll just close by going through some advantages, potential of this approach. The first is that Normally, if you have a competitive inhibitor, you have to go after highly conserved sites. Here you can go after adjacent sites that are going to be less conserved. Number two, if it's a competitive inhibitor, you have to compete with the natural ligand, which is often in a very high concentration, a GTP for RAS, for example, or has incredible affinity for the site. Again, GTP for RAS. Third, with induced proximity, you have the challenge of bringing two things together that don't normally come together. So you really have no template that you're designing to. In the case of sustained proximity, you have a very defined template that you're designing to, a pre-existing macromolecular interface. Number four, in induced proximity, just because you bring two things together doesn't mean you're going to get effector function. There's many cases of bringing a ligase and a substrate together and no ubiquitination happens. You can bring a horse to water, but you can't make it drink, all right? In the case of sustained proximity, usually if you have a high off rate, there's a biological rationale for why that off rate is high. If you slow down that off rate, I think the likelihood that you're going to have a biological consequence is extremely high. And it doesn't take much. If you add as little as one hydrogen bond, 2 to 3 kcal per mole, if you put that in the Gibbs free energy equation, that's potentially a 150-fold effect on off rate. So that's like taking an off rate of 60 seconds and turning it into an off rate of 2.5 hours. And you don't have to think deeply about biology to realize that if you go from a T1/2 of 60 seconds to 2.5 hours, you're probably going to have a problem. Now, I said at the beginning that we're in the fourth wave. This is just tracking FDA approvals of multi-specific drugs. The first one being Enbrel in 1998, which is an Fc fusion of TNF extracellular domain, TNF receptor. And you could see that it's a greater than linear increase over time. 1 in 6 FDA approvals since 2022 to 2024 have been multi-specifics. Half of all multi-specifics have been approved since 2020, so this is really building momentum. But this is the killer. This is clinical trials. And notice the axis on the right is 10x the axis on the left. And they are growing even more steeply. So what we are seeing for the approved molecules is literally the tip of the iceberg. That may not even be a good enough analogy, right? This is a wave that is building and building very rapidly. And I think is going to transform patient care. It's going to give us molecules that do things that we really never thought were possible to achieve with drugs. And we're already seeing that in some of the early multi-specifics. And I would just like to close by saying, you know, one thing I learned at Amgen is to keep the patient at the center. Everything we do, whether it's basic research, you know, when I was a graduate student, I discovered Sec61, the translocation channel. There's now like 2 biotech companies that are developing drugs against Sec61 to control differential secretion of various signaling cytokines and other signaling proteins. Now, I was just out to do that because I was curious about the biology and how cells worked. But everything we do, whether it's basic research, whether it's trying to make drugs, at the end, the patient is at the center. That is why we do life science research. That is why the government funds life science research. It's all about the patient. And all of us, I have people in my family who are dying, who don't have therapeutic options. I imagine that all of you know people that are in a similar situation. And what the past century has taught us is that those unmet needs can be met. It's only a matter of uncovering the knowledge and bringing forward the creativity to make drugs that will treat conditions that we currently think are untreatable. So with that, you know, I leave you to go forth and do great research, and hopefully invent drugs to treat people who currently have no options. Thank you.
Wow, wow, wow. I'm going to invite Dean Rosenblatt to give a token of appreciation to Ray before we start Q&A. Okay, please bear with us for a second. Dean Rosenblatt.
Evocative and educational. Thank you.
Thank you.
I'll get one quick photo here.
Thank
you.
Okay.
Now, time for question and answers. Who has the first question? Go for it. Please.
So I say that with the caveat. So my question is, if you modulate transcription factors, wouldn't that add selective pressure for mutations? And what are possible kind of like repercussions of mutated transcription factors? And I ask that because I'm not a biologist.
Okay, so number one, I'm not talking about mutating transcription factors, but talking about, manipulating their activity.
Yes.
The first point you raised about mutation, there's not really an answer to that, but it's exactly the same problem with every drug, right? If you're talking about cancer. Now, you can use the same approach in immunity, right, and inflammation, and there, mutation is not a concern. The concern is really when you have cancer where you have cells that can multiply and then you can select variants out of that population. But that exact same problem, I don't care what your modality is, what your mechanism is, mutation is always going to be an issue, and it's always something we're going to have to contend with in oncology drugs. Now, the second part of your question was, the first was about mutation.
Oh, it was mainly like, with the estrogen receptor specifically, right? If you have tamoxifen, you have mutants that occur in the estrogen receptor that cause constitutively active estrogen receptor, essentially, that causes metastasis. So it was kind of along those lines of, if you do have a drug that modulates the transcription factor, would you then kind of be in a similar situation? And you already answered it. You said yes.
Well, I think you also asked, though, about might there be undesirable effects of inhibiting a transcription factor. Again, as I said initially, even if you have the perfect drug, that's always going to be an issue because things are repurposed again and again and again in different tissues for different pathways in the body. And that's not going to magically escape that. So, then one has to think about, are there ways? So number one, can you go after a transcription factor that is really limited to the tissue of the disease tissue of interest? And in some cases, for example, there are lineage-specific transcription factors, for example, in the immune system, like GATA3, as a classic example. So you can go after lineage-specific transcription factors, or you could think about, the third type of multi-specific I outlined in the beginning, where you attach some other molecule that has the effect of localizing your drug to where you want it to act. And so increasingly, people are thinking of making antibody drug conjugates where the drug is not necessarily a potent cytotoxic drug, which is what people are currently doing, but is, a much more subtly acting drug, but you just want to localize where it's going to have its impact by targeting it to cells based on cell surface antigens.
Leisure?
Thank you for your wonderful talk. So when I think talk about people try to get into drug discovery for patients, I think we're all for that, but basically it's a funding money issue. Without money, you cannot do any research. That's my comment. My question actually is more specific for your PROTEC technology. If you look for antiviral, you start with like HPV. But if you look at literature for the last 20 years or so, there are not many papers published on PROTEC technology against antiviral. I'm not sure why that is the case, because a lot of anti-cancer. But do you know why that's like lack of drugs or like a lot of research on antiviral for that?
It's a good question. I would say the number of company oncology is a lot of times the answer why, because most of it works in oncology, right, on PROTACs. 50% of the biopharmaceutical budgets of the entire industry are being spent in oncology. So that's just where most of the money goes. So that's part of the answer. The second part is if you're developing a new modality, and there's a lot of risk in doing that, oncology is very popular because number one, you can potentially get fast readouts, especially if people have a rapidly progressing cancer. So you don't have to wait long to see if they're getting worse or you're having a positive treatment effect. And number two, there's less concern about side effect because you're usually testing in people that have no other therapeutic options. So side effects that would be verboten in cardiometabolic medicine are okay in oncology. So, I think for those various reasons, like you don't see PROTACs in cardiometabolic disease either. It's not just infectious disease, virology. So I think there's a lot of, when you have a new modality like that, there's just a lot of focus in going oncology because it's perceived to just be an easier path at the outset.
I get a sense when I went to start sections, people, they say PROTAC will not work for antiviral, but there has to be enough effort to show it doesn't work. Almost like unless you show it works, people say it doesn't work. This is probably standard like for drug discovery, I guess.
I don't see why it wouldn't work, especially given the fact that viruses essentially use PROTAC-like proteins.
Jeff Loeb?
Yeah. Okay, hi, wonderful talk. And thinking, how do we solve problems when we have a target? But the scary thing is that you showed so many companies are just bandwagging on those same targets as a way of de-risking. But then the other challenges, particularly in diseases I study, like brain disorders like Alzheimer's, what if that's the wrong target? And yet you have more and more companies putting all of the resources. So how do we de-risk novel targets? I mean, clearly we're getting better at finding ways of hitting those targets, but how do we de-risk novel targets and how do we find those targets?
Yeah, well, that's why a lot of people keep working on the same targets, right? Because it's hard to do that. So what are the options? So, the main option has been mouse. And as people point out again and again and again, we've cured mice of cancer many, many times over. And a lot of times it just doesn't work in a human.
Brains too, we cure.
Mouse physiology, like if you just consider it a cardiometabolic system, like mouse blood pressure, mouse heart rate, it's so radically different than human that same even with the immune system in mouse. Again, there's very significant differences with the human immune system. And so the model isn't perfect. Now people are talking about, there's been a big shift, like FDA just put out a position paper on this about organoids, using organoids especially for toxicology. Yeah, you can use human tissues in organoids, but if you think about things like obesity, it involves signaling along the gut-brain axis, right? And, as well as adipocyte stores as well. So, you're not going to really recreate that in an organoid, at least not easily. So, again, a lot of inflammatory diseases involve multiple cell types in the inflamed tissue. And again, recreating that is a challenge. So AI, good luck with that. So, at the end of the day, pharmacology is the gold standard, right? You make a molecule and you put it into a human being and it either does what you hope it's going to do or it doesn't. And that's why the minute somebody has a positive signal, on a new target, there's this enormous herding effect. And we saw this with TL1A, with the release of the Prometheus results and inflammatory bowel disease a few years ago. And then all of a sudden everybody needed a TL1A program because it was a new target for that disease. We're seeing that now in oncology with the PD-L1 VEGF bispecific antibodies. Akeso and Summit, showed positive results with theirs, and all of a sudden there's, BMS is laying down hundreds of millions. BioNTech is laying down hundreds of millions. Pfizer is laying down, so pharmacology is the gold standard. And the minute something is shown to work, you can be sure there's going to be multiple programs launched in very short order because it's the only way to know for sure.
All right, we are well past the hour, but before we go, I want to take this opportunity to personally thank Dean Rosenblatt, who helped start this lecture series. As all of you know, he's going to be leaving UIC for greener pastures, better weather in the middle of January. And therefore, this represents the last Dean's Distinguished Lecture under his executive deanship. And therefore, I want to take just a few seconds and personally and on behalf of all of you, thank Dr. Rosenblatt. Next, for the goodies. By habit, don't go to the faculty alumni lounge. There is something else happening there. We made it very convenient for you. Just walk across the street, the floor diagonally, and that's where all the goodies will be, and you can continue to talk to our distinguished lecturer today. Ray, I cannot thank you enough. It was such a fantastic lecture. Thank you very much.